Hey guys, so we finished transcription. We've got this mature transcript, 5' cap, poly A tail, introns have been removed, and now we're ready to do translation. When we do translation, the mRNA, that transcript, codes for amino acids in triplets, in threes. So AUG codes for methionine, and CGU codes for arginine, and GUA codes for valine. And each one of these, right, has a specific amino acid that the three nucleotides code for. Those three nucleotides together that code for a specific amino acid, that's called a codon. And codons are the recipe for proteins. And this is highly conserved among all species of life on Earth. We all do it the same way, whether we're a bacteria, a bread mold, your mom, a goat, whatever, it doesn't matter. If you mess with the code, then you mess with your ability not to make a protein, but to make all of your proteins. Immediately, any organism that started to do that would become non-viable. None of its proteins would work. So this is highly conserved. If you mess with this, you get dead. When we look at these codons, you have four possibilities. You can have an A, a G, a C, or a U at each of those three locations. So that's 4 times 4 times 4, which means 64 different possible combinations. Now, there's only 20 amino acids in life on Earth, so there's some redundancy. We also have to have codes for the stop codons. The start codon actually codes for an amino acid called methionine. It says methionine or start. It should say methionine and start. We have these 3 stop codons. So those 3 stop codons don't code for an amino acid. Everything else on here does. So 61 different codes for 20 amino acids. So there's redundancy. You can see leucine is 6 different codons, all code for leucine. Arginine, has 4 different ones. So there's redundancy in there. So the codons are matched to the amino acids by the help of another molecule called tRNA. And it uses base pair rules to determine which amino acid is next. So we have tRNAs that have UAC on their anticodon, which base pairs with A, U, and G. And you can see they've got the 5', 3' thing going on. And any tRNA that has UAC on it is going to have methionine attached to it. It's going to bring methionine. And if it has GCA on it, it's going to bring arginine. And GCA is going to base pair with this codon of CGU. And if you go back to that table, you'll see that CGU codes for arginine, and so on. So the part on the tRNA that base pairs with the codons are called anticodons. They are antisense. Remember, codons are sense. The transcript is sense. So the anticodon is a triplet of nucleotides that base pairs with the codons. They are on tRNA molecules. These are just 3 different diagrams, actually 4 different diagrams showing tRNA. Of course, the cell part that does this are the ribosomes. And, some people will call them organelles, but really there's big molecular complexes. They're part RNA, part protein, and they assemble proteins. There are 2 parts, there's a small unit, a large unit. You don't have to worry too much about that. When we start to look at the actual process of translation, it can get kind of complicated pretty quickly, but you don't need to worry too much about it. Just understand the role of each molecule. The transcript is the sequence of amino acids that we're going to use, right? It has a triplet, it has a codon, a codon, a codon, a codon, and it says this amino acid, then this amino acid, then this amino acid. We're going to build a chain of amino acids in a particular order based on that mRNA transcript. tRNA's job is to bring the amino acids and make sure that the appropriate amino acid is brought at the appropriate time. The ribosome's job is to facilitate the whole thing. It really makes it all happen. It holds it all together. It holds on to the growing polypeptide. Codons are on the mRNA, always. Anticodons are how the tRNA know which amino acid comes next. So here's my transcript. The tRNA brings an amino acid. This is the ribosome in the background here. The next tRNA is going to base pair, right? We get these base pair. We're just following base pairing rules here. And we're going to grow this polypeptide as we go. Eventually, we've got a long chain. Eventually, we hit a stop codon. There's a protein that binds to that, causes the whole thing to fly apart, and we're all done. So when we build a polypeptide, there's a couple of terms you need to know: N-terminus and C-terminus. And really, this is just about the assembly of that polypeptide. The amino acids, right? Remember, amine is like this. We've got an R group here, and there's 20 different ones in nature, and that's what makes, methionine, glutamate, whatever. And then we've got a carboxylic acid group here. So we have this amine group and we have this acid group. And when we go to attach another one, it's going to take out this hydroxyl and it's going to bind like this. And so I'm going to end up with another carbon, and etc. And so I'm going to get this NCC, NCC, NCC repeating chain, right? In each of these. So that's going to make up the backbone of the polypeptide. So the N-terminus is the first one we put down, right? The first amino acid we put down is going to be the N-terminus. And then the far end of the molecule, the C-terminus, will be the other end. We've talked about this before. Basically, we're talking about secretory pathway. It is the sequence of amino acids that tells the ribosome, stop building this protein until you are attached to the ER and you can put it through that portal, right? So we build it into the ER when that signal set of amino acids is present. It doesn't have to be secreted. It could be going to the lysosome or to the mitochondria or anything that is membrane related. It's going to have to go through this process. Now, bacteria don't have organelles. They don't have an ER. How are they going to do secretion? Pause the video and see what you can figure out. Hopefully you figured out that, well, the simplest explanation is they build it directly into the membrane. So it goes right outside immediately. So it's got to be done then. We can't do any processing after that. It's outside the cell. It's gone. It's doing its thing. But bacteria have to do this. I don't know if you remember, we did the digestion lab. We had those bacteria that digested the starch on the petri dishes. But they secrete those digestive enzymes. And when they secrete them, they assemble them outside of the cell through one of these portals. And they are fully functioning when they're out there. So this diagram sums up all the parts you need to know. Maybe one you don't. I don't know if you need to know amino acyl tRNA synthetase. That's not that big a deal. But its job is basically to recharge this tRNA. But going all the way to the beginning, right? DNA, we're going to use RNA polymerase and transcription factors to build that transcript. We're going to remove introns. We're going to put a 5' cap on. We're going to put a poly-A tail. That's what makes it a mature RNA. Until then, it's called a pre-mRNA sometimes, or a mature transcript. The two subunits of the ribosome are going to assemble, and they're going to use that transcript. And some tRNA are going to bring amino acids, and they're going to start assembling that polypeptide. And like I said, see this tRNA, it's already added its amino acid to the growing chain. And so it's going to go back to this enzyme here and get recharged with another amino acid so it can go do it again. All right, when we look at the transcriptional unit, we look at what we sometimes call a gene. First, we're just looking at the template strand. We got the stretch of it that's going to code for the protein. It's got a promoter sequence. It's got an enhancer sequence upstream a ways, and that's technically part of the gene too. With the appropriate transcription factors, RNA polymerase will be summoned, and it will start at one point, and it'll end at one point, and that's the stretch that gets made into that primary transcript. Anyway, so of course we're going to take out introns, leave in the exons. This is our start codon, right? AUG is our start codon on the mRNA. You don't call it codons on DNA, only on messenger RNA, only on the transcript. So TAC, though, that's the template strand that's going to code for AUG. So that's the place we're going to start translation. And we're going to stop translation at the stop codon. So we have these two regions that are called UTRs, and those are untranslated regions. Basically, it's a little bit after the stop and a little bit before the start, where we're going to make the RNA, but we're not going to use that to make a protein. Not that big a deal. It does come into play a little bit later on when we talk about regulation. So we get this pre-mRNA. We remove introns. We stick a 5' cap on it. We stick a poly-A tail on it. We've got introns removed. We've got a mature transcript. In prokaryotes, protein synthesis is a little bit different. It's not a huge deal. There's just really one main thing you need to know. But we'll go through some of the differences. First of all, DNA is in the cytoplasm in bacteria. There's no nucleus. Their chromosomes are a different shape, not really relevant to protein synthesis. The DNA cannot be blocked by proteins. These histone proteins, this is part of how we turn off like big areas of the genome, by wrapping it tightly around these histones so that the transcription factors can't get to them. There are no introns. This is the big one. Bacteria don't have introns. So they don't do any of that processing. They still do a 5' cap. It's a different molecule, polyethyl, but they do not remove introns. And of course we do. So transcription and translation can happen simultaneously in bacteria because there's no boundary. We're not going from the nucleus to the cytoplasm doing transcription and then translation. We can start doing transcription, and as that RNA comes out, ribosomes are going to grab onto it and start translating it right away. And that's what these pictures are showing. It's simultaneous. Not that big a deal, but it should make sense to you. Without that physical difference, it actually makes it so they get proteins a lot faster than we do. So that's translation. You got a little bit of an assignment to do. It's just a series of slides that you're going to rearrange and label. If you have questions about anything, please shoot me an email. We can jump into a Google Meet, no problem. Hope you guys are doing well. Take care.